Chapter 4 deals with the anatomy and histology of muscle. And we know fundamentally there are three different kinds of muscle. Two muscles are classed as striated muscle because of the banding pattern, and the third type of muscle is smooth muscle. First slide illustrates the fundamental histology or structure of a skeletal muscle fiber, which is really a skeletal muscle cell. And what you're seeing is surrounding the muscle cell is a cell membrane called the sarcolemma, and basically that individual muscle cell is surrounded by a connective tissue coat called the endomysium. Surrounding several and running between adjacent muscle cells is a paramysium, and surrounding the entire muscle will be an epimysium. So there are three different layers of connective tissue that surround different elements of the entire muscle, separate muscle cells from one another, and engulf individual muscle cell by surrounding the sarcolemma. So the other thing you're seeing is that the cytoplasm of a striated, in this case skeletal muscle cell, is dominated by a series of parallel myofibrils that contain parallel arrays of actin and myosin filaments organized into sarcomeres. The other point that we can make on here is you see that one of the other characteristics of a skeletal muscle cell is multiple peripherally situated nuclei, one of which is illustrated on this slide. And again, when we use that criterion to distinguish a skeletal muscle cell from a cardiac muscle cell. So again, here is the summary of the connective tissue coverings. The epimysium is dense connective tissue that surrounds the entire muscle. The paramysium is a thinner sept of connective tissue that extends between the epi and the endomysium and surrounds all of the individual muscle fibers. And then the endomysium basically surrounds each individual muscle fiber or muscle cell and is in direct contact with that muscle's sarcolemma. The next slide illustrates the detailed histology and structure of individual sarcomeres. Note first that by definition, a sarcomere corresponds to the distance between a pair of Z lines. Attaching to the Z line on either side of adjacent sarcomeres are the thin filaments formed by F-actin that form collectively the I-band. So the I-band consists exclusively of the actin-thin filaments on either side of the Z line. In the middle of the sarcomere is the A-band, and the A-band consists of exclusively myosin filaments that are overlapping basically with the actin filaments in the middle or central portion of the sarcomere. Basically, where the actin filaments do not overlap the myosin filaments, that region literally in the central part of the sarcomere forms the H-band. And the line that exactly marks the middle of the sarcomere is known as the M-band. It's known as the M-band. Note that when the sarcomere shortens during contraction, that the length of the A-band will not change because it simply corresponds to the length of the myosin filaments, but the I-band and the H-band will shorten because the sarcomeres will decrease their length, and obviously it'll move the myosin filaments closer to the Z line on either side, shortening the I-band. And in addition, the actin filaments on either side of the H-band will move closer together, shortening the A-band as well. So obviously the bottom line is during muscle contraction, the sarcomeres shorten as the Z lines move closer together. The next slide shows a histologic section of a typical skeletal muscle cell stained with hematoxylin and eosin. And what's readily apparent in this preparation is the distinct banding pattern of I and A bands that make up sarcomeres. For example, you can see distinctly how well the dark myosin-containing A bands stain in the middle of each sarcomere, basically there at the arrows, whereas the lighter staining I bands are situated at the arrowheads. And in addition, you can make out a faint dark line in the middle of that I band, which constitutes the Z line. In addition, because they're very characteristic, skeletal muscle cells are multinucleated, and you're seeing an example of a peripheral muscle cell nucleus that's situated on the right side of this muscle cell. The next chart makes a comparison between red versus white skeletal muscle fibers. And basically, their color terminology comes from their degree of myoglobin content. Red fibers have a high myoglobin content. White fibers have a low myoglobin content. Red fibers are specialized for slow conduction. They generally exhibit decreased ATPase activity and utilize aerobic metabolism through mitochondria as their energy source. White fibers are specialized for fast contraction. They show increased ATPase activity and utilize anaerobic glycolysis as opposed to aerobic glycolysis, and as a result show a decreased mitochondrial content. Red fibers are specialized for slow posture-maintaining muscles, whereas white fibers are associated with fast, short-term skilled actions that are associated with sprinters, lower limb muscles, ocular muscles, and muscles that are intrinsic to the hand. There are two additional proteins that form structural functions within sarcomeres of skeletal muscle. Nebulin is a scaffolding protein that binds to the actin filaments along their entire length and also inserts into the Z-line, so it stabilizes basically the I-band. Titin is a very large scaffolding molecule that binds to myosin. It is anchored to the middle of the A-band at the M-line and also extends along the free end of the myosin molecule to the Z-line. Skeletal muscle has a limited regenerative ability, and its stem cells, are known as satellite cells, are situated just outside the plasma membrane that are capable of differentiating and forming new myoblasts. And one of the characteristic features of muscular dystrophies is that they tend to deplete the pool of satellite cells. Next, let's look at a useful chart that compares the overall structure and histology and even fine structural features that will allow us to distinguish skeletal muscle, which we've talked about, from cardiac and smooth muscle. First point we want to make, as we alluded to earlier, is that skeletal and cardiac muscle are striated, meaning that they consist of a series of myofibrils containing sarcomeres. Other than that, skeletal muscle cells are generally unbranched, whereas cardiac muscle cells are highly branched. So in terms of the over-sarcomere structure, it's difficult on that basis alone to distinguish a skeletal muscle sarcomere from a cardiac muscle sarcomere. So we have to use other criteria. Number one is, as we've seen, skeletal muscle cells tend to be multinucleated, and their skeletal muscle nuclei are situated adjacent to the plasma lemma on the outside of the muscle cell. In contrast, cardiac muscle cells have a single centrally placed nucleus. Smooth muscle cells are non-striated. They lack Z-lines, but in their place they have dense bodies, which are attachments for the actin and myosin filaments. And like cardiac muscle cells, smooth muscle cells have a single nucleus, but smooth muscle cells tend to be more fusiform in shape rather than the elongate branched cardiac muscle cell. The T-tubules are slightly different anatomically as well. The T-tubules form extensive contact with the adjacent sarcoplasmic reticulum. And in skeletal muscle, the T-tubules and the cisternae of the sarcoplasmic reticulum form triads that are distinctly located at the AI band junction. Cardiac muscle T-tubules are situated near the Z-line, but the sarcoplasmic reticulum tends to be less well developed and typically only forms a dyadic relationship with the T-tubule. T-tubules are absent in smooth muscle, and they themselves have only a limited sarcoplasmic reticulum. The other real ultrastructural feature which enables us to easily distinguish cardiac muscle cells from skeletal muscle cells are the presence of specific junctional complexes known as intercalated discs by light microscopy that separate and or join individual cardiac muscle cells. Such cell junctions are absent in skeletal muscle cells, but as we said, are going to be a unique feature of just cardiac muscle cells. So these junctional complexes are a combination of desmosome-like contacts known as a fascia adherens interspersed with intervening gap junctions. Smooth muscle cells lack the desmosomal relationships between their additional cells, but smooth muscle cells, like cardiac muscle cells, are joined by gap junctions. The next schematic diagram shows an array of myofibrils containing parts of 3 sarcomeres and also illustrates the relationship of the sarcoplasmic reticulum to adjacent 2 tubules in a triad. Note that this close relationship is essential for spreading a wave of depolarization around and between the adjacent myofibrils and activating the release of calcium from the sarcoplasmic reticulum of a striated muscle cell.